Okay. Hi everyone, I'm Trisha.
And I'm Dafina.
And for our gear project this year, we did a proteomic analysis of nerve regeneration. So the main objective of our research was to analyze both injured and uninjured nerve samples and then determine the key molecular pathways that changed, which is like the presence of certain proteins. And then we would use that information to get a better understanding of the regeneration process. So, for a little introduction, with the traumatic nerve injury, a nerve is severed into two pieces, and that leaves a proximal end that's still connected to the central nervous system, so like the brain and spinal cord, and then a distal end, which is completely cut off from the central nervous system. And in order to repair this nerve injury, there's a process called the autograft procedure, where a portion of a nervous tissue from either another part of the body or a donor is inserted into the nerve injury, into the site of injury. And then when this occurs, there are many cellular pathways that change because of the regeneration process that follows. But this is not well understood in the community. So we're doing a proteomic analysis to basically analyze the presence of different proteins following nerve injury in order to understand how they affect regeneration.
Right. So to go more into depth with our methods and how we completed this analysis, firstly, sorry, Peter made a noise. Firstly, we used a rat. We actually used rat models to basically replicate this nerve injury. And what we did is we extracted a tissue sample from the proximal end of the hind leg from a rat 5 days after its injury. And I guess we can go ahead and show the images of the autograph, stretch, and control, since she just mentioned it. So we have the autograph where they inserted basically they took a nerve from somewhere else, inserted it to basically repair the nerve and have it connect from both ends. This is usually if there's like a large gap. And then we have the uninjured control, which is what we're analyzing. We're comparing it to the autograph. And this is basically our control, like without any changes or repair done on it. And then we also have the stretched, which is the proposed solution from the Shaw lab. And this will go more into detail with later. But basically, they stretch the tissue in order to fix this gap instead of using an autograft. So if we go back to the methods, we can see a flow chart. And taking we basically extract a tissue sample and from the tissues, we obtained two groups, which is the autograft and uninjured control that we're examining for our analysis. From this, we can then perform mass spectrometry, which if you're not familiar with, basically what it does, it's able to detect and separate ions based on their masses. So since it's able to do this, it's actually able to differentiate proteins that are present in samples. And then from there, we can identify what exactly the tissue is made up of in terms of proteins. So from the mass spectrometry, we're able to get a whole list of proteins that are present in the samples. And basically, it gives us like these universally known IDs that are associated to proteins throughout the scientific community. And from this, we can see, we can get the samples we can get the IDs that are significantly different between the groups. From this, we use David, which is an online database, which is able to then cluster the proteins and organize them based on how similar they are and kind of group them automatically for us. But from there, we then want to manually cluster them and further refine what we got into functional groups that we determined to be significant, which we'll go more into later. From this, we can perform a literature review and comparison of the autographed and uninjured control samples so that we can further understand the results that we got and further analyze what exactly is going on 5 days after injury when we compare autographed and uninjured control samples.
Yeah, so moving on to results, in this first figure right here, we have a graph that gives us a distribution of the proteins with significant differences before and after nerve injury. So the x-axis is a list of all of the proteins. There were about 150 of them. And then the y-axis represents the significance value. So all of these proteins here at the top are involved in ribosomal and translational processes. So this basically tells us that in the regeneration process, those proteins are highly upregulated in comparison to before injury. So like there's an upregulation of these proteins during the regeneration process.
Right. And so to go more into table one, which furthers this analysis, basically this is the final resulting clustering that we got from automatically and manually organizing the proteins from our control and injured samples. And as we can see, we kind of put a count of how many were in each group that we determined. And from these results, we can see that there's a high number of bioenergetics, protein folding, and degradation pathways that are changing post-injury from the tissue samples that we obtained.
And then for future directions moving forward this summer, we plan to develop a Python algorithm that would let us sort these proteins in a more efficient way, because we had to like manually look up each protein and their function and then determine what cluster out of these it would fit into. But since the database is already out there with David, we want to develop a program that could basically sort the proteins into these clusters for us, so that could save us all the time it took.
Another thing that we want to do is compare these outcomes that we obtained with those from with other regenerative strategies, predominantly the stretch solution that was developed by the Shaw Lab, so we can further understand how different they are, how much better or worse they are at regeneration, and also so we can further analyze for future publication of a paper in the nearby future. Another thing, lastly, that we would like to do as well is view later time points of regeneration. So we took tissue samples 5 days post-injury, but I think we would like to ideally look at later time points, like after 5 days, 10 to 20 to more, since I think, in real life, you're going to want to understand processes in a longer time frame and see what changes are exactly occurring to further understand what regeneration processes are going down and what proteins are being up or down regulated. But yeah, so in conclusion, this is our poster. We would like to thank Professor Samir Shah, who is here today, for his guidance, as well as Neha, the graduate student, whose work greatly helped us, and talking with her, it was great. A lot of guidance and support throughout the project. We'd also like to thank Ved, Alejandra, and the rest of the GEAR community for their constant support and being a community. And yeah, thank you all for being here today and listening to us.
Thank you.